us see post-fertilization changes. That is, after fertilization, what are all the changes takes place? See, first of all, the ovary change into fruit. Ovule changed into seed. Ovary wall forms the pericarp or wall of the fruit. Integument change into seed coat. Outer integument, it forms the outer seed coat, that is called as testa. Inner integument, it forms the inner seed coat, it is called tegmen. New cellus degenerates, but sometimes it is present in the form of perisperm. This we have seen in previous classes. Next, the egg cell becomes embryo. Synergids and antipodals degenerate. Then hilum of the ovule. You can see a scar in the seed, in the mocha and the பாசிப்பயிரில் பார்த்தீங்கன்னா வெள்ளையா ஒரு இது தெரியும். அதுதான் hilum. That is the hilum of seed. That seems to be a scar on seed. Next, funiculus of ovule. It forms the stalk of the seed. That is, the seed attached with the help of funiculus to the fruit. That is called, stalk of a seed which formed by funiculus. Next, micropyle. The micropyle is present in the seed which absorbs water, and also it is the path for the entry of oxygen also at the time of germination. Then chalaza of ovule. It forms the chalaza of seed. Next, we will see the ploidy level in different parts of plant. See, these are sporophyte. Zygote, embryo, radicle, plumule, cotyledon, nucellus, integument, microspore mother cell, megaspore mother cell, ovary wall, fruit wall, carpel, sepal, petal, stamen, leaf, root, stem. All these are in which condition? No? Diploid condition. Then the gametophyte you can see, microspore or otherwise pollen grain. Then tube cell, generative cell, male gamete, female gamete, megaspore, embryo sac, synergids, antipodals, egg cells, and two polar nuclei. All these are in haploid condition. Let us see the life cycle of angiosperm. See, angiospermic plant, it has a flower which has carpel and stamen. Carpel otherwise called as megasporophyll, stamen microsporophyll. Carpel has ovule, stamen has pollen sac. That is, the ovule is megasporangium, pollen sac microsporangium. Then megaspore mother cell, microspore mother cell. This megaspore mother cell produce megaspore, and these megaspore develop into female gametophyte, that is embryo sac. This microspore mother cell undergoes reduction, division, meiosis, and form microspores, that is the pollen grains. And this is the male gametophyte, and these forms two male gametes. And in the female gametophyte, that is in the embryo sac, secondary nucleus is present, and also female gamete egg is present. And then these male gamete and the egg fuses. That process is called as fertilization or syngamy. Then form zygote. The zygote develops into embryo. Then again it grows into adult plant, produce seed, again germinate and form another plant. Then at the same time, secondary nucleus fuse with the second male gamete, and this fusion is called as triple fusion, and this forms what? Endosperm. We will see structure of monocot seed. The seed of paddy is called as caryopsis. The seed of paddy is called as what? Caryopsis. Caryopsis. And the seed is enclosed in a brownish husk. Seed it is enclosed by means of brownish husk. And this consists of the glooms. In how many rows? 2 rows. 2 rows of glooms. The next, seed coat. How is the seed coat? No, it is brownish, membranous, and adhere closely to the grain. Next, endosperm. This endosperm forms the bulk of the grain, and the grain full endosperm. How the endosperm is separated from the embryo? By means of epithelium. The endosperm is separated from the embryo by means of what? Epithelium. The embryo is small. The embryo of monocot is very small. Next, That is, the embryo is, then the cotyledon is present in the form of shield. One cotyledon in the form of what? Shield. That is called scutellum. That is called as what? Scutellum. The next one is, then where this scutellum is found? Lateral to the embryonal axis. It is found lateral to the, which axis? Embryonal axis. Lateral to embryonal axis. Next one we will see apomixis. Apomixis. Before that, you must know what is amphimixis. Amphimixis. What is meant by amphimixis? Fusion of male gamete with the female gamete, that is syngamy, or fertilization, that is called as amphimixis. Then what is apomixis? That is, first we will see who introduced the term apomixis. Introduced by Winkler. Introduced by Winkler in the year 1908. The year 1908. And this apomixis is substitution to sexual reproduction. It is substitution to what reproduction? Sexual reproduction. The next one is, then what is the difference between amphimixis and apomixis? In amphimixis, there is meiosis and syngamy. But in apomixis, there is no meiosis and no syngamy. That is the difference between amphimixis and apomixis. Now we'll see the types of apomixis. Apomixis, two types are there. First one is recurrent. First one is what? Recurrent. Second one is non-recurrent. First one is recurrent. Second one is non-recurrent. Under recurrent, there are two types. Vegetative reproduction, and another type is agamospermy. Agamospermy. Then what is non-recurrent? This is haploid. What develops? Haploid embryo sac. Haploid embryo sac developed after what division? Meiotic division. Haploid embryo sac developed after meiotic division and develop into embryo without fertilization. That is known as non-recurrent apomixis. I'll repeat again. Haploid embryo sac develop after what division? Meiotic division. But after that, embryo is formed without fertilization. So these are the two types of apomixis. First you will see recurrent. Recurrent apomixis. In that, two types we have seen: vegetative reproduction and agamospermy. Vegetative reproduction already we have seen in previous chapters. Now we will see agamospermy. What is meant by agamospermy? That is embryo develop without meiosis and syngamy. That is called as agamospermy. That is embryo develop without meiosis and syngamy. Then there are three types in this. Let us see the first one. Adventive embryony. Adventive embryony. What is adventive embryony? The embryo developed from diploid nucellus or integument. See, listen. The embryo developed from diploid nucellus or integument. This is otherwise called as sporophytic budding. This is otherwise called as sporophytic budding. And here what is absent? No, gametophyte is absent. What is absent? Gametophyte is absent. Gametophyte is absent. Example for this is citrus Mangifera. Mangifera. So first type over. Second type is diplospory. Second type is diplospory. Now what is diplospory? That is, it is otherwise called as diplospory is otherwise called as generative apospory. The other name is generative apospory. Here, that is the megaspore. The megaspore mother cell develop diploid megaspore without regular meiotic division. Without regular meiotic division, this is called as what? Diplospory. Example for this, Eupatorium and Parthenium. Sorry, not Parthenium, Area. Eupatorium and Area. The next third type is apospory. Third type is what? Apospory. Here, the megaspore mother cell gives 4 haploid megaspore. What happened to this 4 haploid megaspore? It disappears. Then how the embryo develops? How the embryo sac develops? It develops from new cellus. And this is called as apospory. The other name for this apospory is somatic apospory. It is otherwise called as somatic apospory. Example, Hieracium and Parthenium. Example, Hieracium and Parthenium.